Well, good afternoon, and thanks for coming back. I know it was a short coffee break. So we're going to reconvene. In our next series of speakers, we're going to hold the Q&A until the panel discussion. So they'll each be giving a talk, and then we'll have Q&A at the panel. So let me now introduce Dr. Steve O'Brien. He is NSU's only member of the National Academy of Sciences. We're very proud of that and proud of Steve. He has a long tenure, has a 25-year tenure as chief of the Laboratory of Genomic Diversity at the National Cancer Institute, National Institute of Health. He's also the chief science officer of the Theodore Dubzhansky Center for Genome Informatics in St. Petersburg, Russia, since 2011. I think he's recently retired from that, but keeps an advisory role. We're proud to have him as part of our faculty. He does great research in various areas, human and comparative genomics. And you can read all these things here. He works on cheetahs, lions, and tigers, no bears, pandas, humpback whales, white and black rhinoceros, and he is not adverse to corals. So, Steve, welcome, and please join me up here, and we'll hook you up.
You haven't read my bear papers.
I haven't read the bear papers.
Okay. Oh. Well, welcome to the Ocean Science Symposium here at NOVA. Okay. The subject area brings me to think of a few muses. First of all, it's hard to think of oceans without thinking of evolutionary biology, since they all started there, and we all started there some time ago. And that evolutionary context makes me remind that there's three big dominations of the Earth. One was the trilobites, which you can only see in the Smithsonian, the dinosaurs, which you can see in Steven Spielberg movies, and ourselves, which are illustrated right here by some of our early grandparents. Now, as we watch the domination of the Earth by now over several billion people on the Earth of humans, it's important to think that when we talk about oceans, particularly oceans that we're studying here, Australia, and around the world, it's hard not to be thinking conservation, because there's so many species there that are threatened by ourselves. And when you say conservation, this doesn't just mean genetics, okay? In fact, it means lots of things. It means ecology, human imprints, agriculture, economics, tourism, animal and plant behavior, veterinary medicine, natural history, and finally, the latest thing is genetic inferences and what we can learn about species and perhaps help with managing them. Now, about genetics, when conservation first began, well, in my mind, it kind of started 100 years ago when these guys, John Wesley Powell, John Muir, and Teddy Roosevelt camped underneath Hapdone, and they thought, well, maybe these areas are worth saving. None of these guys were geneticists. They didn't talk about genetics at all. Genetics started really in the 60s and 70s when the technologies came to realize that there were consequences associated with nearly going extinct. One of the poster examples of this is the African cheetah, which my friends Mitch Bush, Dave Wilt, and I studied back then and discovered with the methods that were available to us at the time that the cheetah was very unusual in that it didn't like to breed in captivity like other animals, lions and tigers and horses and hoofstock bred like rabbits, but not cheetahs. They had a lot of trouble, and the reason was they had an unusual genome and had 90 to 99% of the variation, the natural variation, which is the stuff of evolution and adaptation, was gone in the cheetahs because they had descended from a near extinction event we now know was about 10,000 years ago. And these are just a list of the different methods that we applied to the cheetah over the next 20 or 30 years. Most recently, the whole genome sequence era allowed us to sequence the genome, and this guy, Pasha Dobrynin, who was the first author, among other things, he discovered that whole genome sequence diversity, the 10 million variants, was lower in cheetahs than in the Tassie devil, the Amur tiger, or many of the other species. The highest that we saw was in a cat running around St. Petersburg whose name was Boris. The cheetah also told us some lessons about what you can learn with whole genome sequencing, such as the fact that the cheetahs were inbred led to some congenital abnormalities, such as a high frequency of abnormal spermatozoa. This is a picture of some of the bad sperm you see in cheetahs. This one has 3 heads, this 2. This is a big head. This is a little head. This is a rather rare cheetah sperm. It's a normal one. When the sequence of the cheetah's genome was done by these guys right here, an interesting gene that we know in humans and in mice has a big effect in sperm development. One of the primary drivers of it had 5 constitutive damaging mutation which totally destroyed it. So we now know the genetic basis, a CRISPR target, if you will, for cheetahs that we can basically go after to deal with something like the genetic consequences of inbreeding. This was our first lesson. It wasn't the only one. The snow leopard has a mutation in the EGLN1 gene. That means nothing to you people, but the snow leopard lives high elevations in the Amur Mountains and in the Himalayas, as do the Tibetan people. The Tibetan people have the same mutation. That allows them to survive oxygen depletion. So do the horses in the Andean Mountains in South America. So genetics is starting to give functional association to some of the genes and some of the phenotypes that we've been looking at, watching, and trying to understand for some time. Genetics has also been involved in action, such as the things that Dr. Hughes talked about with the corals. And one of the examples that I'll mention to you right now took place not 20 miles away from here in the Everglades and in the Big Cypress Swamp. It's the case of the severely inbred Florida panther, a tiny relic population subspecies of pumas or mountain lions or cougars that survived in the 1960s and 70s as a tiny population or to 20 or 30 animals. It basically lives in the alligator and mosquito-infested Big Cypress Swamp, not too far from Hollywood and Fort Lauderdale. And this population was tracked by U.S. Fish and Wildlife, by Florida Game and Fish, and by people in helicopters, such as Terry described a few minutes ago, who monitored 20 or 30 animals until the demographic modelers said, this population is dead. It's going to be gone if we don't do anything. So what they decided to do was to reintroduce gene flow into Florida, into the Florida panther habitat from Texas cougars, a population that had an overlapping range with the Florida panther less than 150 years ago. So 8 cougars were brought into the Florida habitat in 1995. And 20 years later, the results of that experiment we now know of. The animals were released. 8 of them actually were successful in breeding. But before I show you the results of that, let me explain that there was a lot of problems with the Florida panthers because it was so inbred. It had a high frequency, more than the cheetah, of malformed sperm. 50% of the animals had less than 2 testicles, the male cubs, 0 or 1. Heart murmurs, atrial septum defects, holes in the heart that were lethal, tail crooks, high infectious disease load. Everything about this population said, needs some help. So with the introduction, what happened was the population of Florida panthers in 1995 started to rise up. There were 15 offspring from the first F1 of the 5 Texas females, but within the next 10, 15 years, there were almost 600 cubs produced. It was a huge increase in animals. The animals tripled in their density. They tripled their numbers. They went into habitat the ecologists said they couldn't go to. They jumped out of trees faster than the original canonical animals. And all the correlates of inbreeding went away. This is a success story for genetics. Now it's at the stage where there's so many Florida panthers, they're wandering into the Walmart parking lots in Naples and in places close to them because they've run out of habitat. My recommendation to these people and to these animals is, well, why don't we put it in some of the other ranges? There's 4 or 5 states in the former Florida panther range. Let's put some back there. We're talking about that now. I think it's going to happen. Another example, and there's 4 or 5 examples in my talk that I'll talk about, has to do with the great white shark, the one we all fear because of Steven Spielberg. But thanks to Guy Harvey and Mahmoud Shivi, who's sitting right here in the front row. Guy Harvey's an artist. He painted this thing up here. He also painted the mural in the Fort Lauderdale airport. He is a sponsor, and his name is on this building. They have funded the whole genome sequence of a great white shark, an animal that lives to be very old, 70 years. It has a genome almost 50% bigger than the human genome, which is about 3 gigabytes. A couple months ago, they published a number of fascinating findings about the great white shark, and among them were that they had a long evolutionary lifespan, and there were some genes that were discovered that may have influenced that. In addition to that, they have a wound healing that seems to be better than other species, and there were some genes about that. And there were some genes discovered that are involved in something called genome stability. The shark is a long-lived critter. It's been around for several hundred million years. It's had a lot of evolutionary time to deal with being one of the biggest predators in the sea. And it has accumulated mutations that we need to mine and understand better. And this was the first step in doing that. So the conclusions was that the genome size is really not driven by the abnormal whole genome duplication, but rather a proliferation of internal repeats, a number of genes involved in maintaining genome stability, genes involved in maintenance of telomeres, which are indicators of aging, and they have limited numbers of olfactory receptors or smell, but a complete set of VNS genes for olfaction. There are other things that genomics has been used for, and as I said, I'm going to give you about 3 or 4 examples. Forensics and conservation is one that has been applied unexpectedly to a species we didn't think we needed, which was the white and black rhinoceroses, which live in southern Africa. These species were threatened by overhunting for a long time, but they came back by protection through a man named Ian Player, who was related to, first order brother of Gary Player, the golfer. Ian Player set up a conservation program where the white rhinoceros and the black rhinoceros were brought back to a few thousand in southern Africa. But then what happened is the horns became very valuable, particularly in East Asia. They became valuable as aphrodisiacs, as treatments for cancers, as trophies, and they started to gather a developed, organized criminal network, well, again with helicopters and automatic weapons, to wipe out these animals. In 2007, there were 13 animals poached in southern Africa. That went up precipitously in the last 10 years until it's now over 1,400. This is a huge increase in illegal hunting. And illegal hunting and poaching was traditionally punished when you caught somebody by a slap on the hand. Maybe a night in jail, maybe a $500 charge. No more, because of Cindy Harper and Alan Guthrie. Cindy is a veterinary student and geneticist at Undistaport Medical University. She was offended to know that some of her native South African species were being exploited and extirpated. And so she decided to use her knowledge of genetics and genomics to build a forensic identification for every single rhino in South Africa. She set up a database called RODIS, which stands for the Rhino DNA Information System. You'll recognize this as sounding a little bit like CODIS, which is the DNA information system for the FBI here in the United States. She collected samples from hundreds, then thousands of rhinos across Africa by basically training the rangers to collect materials in a course where they became shall I say, certified as being capable of collecting these individuals. They trained them, the police investigators, the field rangers, veterinarians, prosecutors, to use rhino DNA evidence to collect DNA samples. In the last 8 years, 400 people have been trained. Every day, 5 or 6 crime specimens come in to Cindy's laboratory, which now has 10,000 specimens in her database, all of which have been typed with a number of short tandem repeats, which were specifically mined from the whole genome sequence of the right rhino run at the Broad Institute in Massachusetts. But Cindy took advantage of it and turned that into a diagnostic forensic identification. What she did was she received this many forensications to Rhodes in the last several years, over 1,000 in 2014, 15, and 16, and so forth. They were pieces of material that were found on poachers that were stopped at Heathrow Airport, at Hong Kong Airport, at Johannesburg Airport, in Land Rovers riding across Mozambique, across Kruger Park, and they had blood stains on them, they had horns, they had all kinds of things, and they always used to say, I don't know what this is. It's not a rhino horn. I think it's just a horn off of some, dick or something like that. Well, with the DNA identification, she was able to show, and it was summarized earlier this year in a paper in Current Biology by Cindy Harper and many of her colleagues. I was her PhD mentor, and basically it turned out that these were individual prosecutions where a number of horns or horns were matched to carcasses. So what they were doing was taking crime scenes, which are poached animals, collecting the samples, delivering it to the laboratory, genotyping the individual identification, and then waiting for somebody to catch the poacher. When they caught a poacher at an airport or in a car, they ran the DNA type, and then they went to the database and they said, who does this poacher's stuff match? And in about half the cases, they were able to identify the specific rhino, the date, and the place. DNA evidence, same thing that puts people in jail today for rapes and homicide, was being applied to the poaching crisis in South Africa. This is the top 6 or 7 cases that were featured in the paper. There's actually 50 that have been successful. And the sentences they get are not 4 years or 8 years, they're 10 or 15. A few more minutes, Bernard. Please. All right, domestication has been called the lever that moves civilization. It took place 10,000 years ago, and what happened was many species, dogs, cats, cattle, farm animals, crops were all brought up and essentially domesticated in the Middle East or the Far East or a place called the Fertile Crescent. And in that particular time, we now think that this is the beginning of human domestication as well. Well, back in the 1960s, an individual in Russia, in Novosibirsk, named Dmitry what was Dmitri's last name? Yes, Belayev. And Ludmila, his assistant, decided that they wanted to reproduce the domestication process by taking wild red foxes, or a variant called silver foxes, and to breed them in captivity so that some of them would be so tame they would be happy to be very close to you, while others, when you came close to them, they would hand you your rear end. So that he had after 50 years, there were 2 separate lines. And what happened was, a young woman named Anna Kukova saw this and decided, how are we going to get at the genetics of this domestication process? And she last year published a whole genome sequence with myself and others where she was able to identify the genes that were involved in domestication. And she found that the gene that was the most important was a gene called SORCS1, a gene that codes for trafficking protein of a glutamate receptors. That may not mean much to you, but it's one of the genes that's also involved in domestication of domestic dogs. This is Dr. Belyaev, who died several years ago, in a statue with Anna Kukakova in Novosibirsk. And it basically is a day I happened to be there too, so we took the picture of all three of us. And that basically was the beginning of uncovering the genes involved in domestication of new species as well as old species. Restoration, we've talked about the restoration of the Florida panther. There's also talk about doing this species, which is the Asian tiger. And I'll just say in 1 minute that the tiger subspecies was a confusing mess until Yuli Seal and Peter Jackson came to myself about 15 years ago, and they said, can you help us with your molecular tools figure out how to tell the difference between a Sumatran or an Amur or a Bengal tiger with your molecular tools? The person that did that is sitting in the front row, Soo-Jen Lo. She was a graduate student. Raise your hand, Soo-Jen, so you can see it. She has led the field in identifying the genetic differentiation between the tigers that are still alive, the Amur tiger, the Indochina, Sumatran, and Bengal, and the ones that went extinct, the Caspian, South China, Javan, and Balik. And she did it by doing genetic studies on tigers that were collected across the range and then measuring them according to their geographic position. This year was published a whole genome sequence of a number of tigers. I think it was about 25 or 30 that basically reinforced the subspecies definition with her Chinese students, Lu Chen Liu and Yi Sun. Where are you? Sun, he's in the back there. Go talk to him. It's an amazing study that basically took 32 modern species and basically were able to come up with a differentiation between it. This meant that there were now not 8, but 9 tiger species, one of which, the Malayan tiger, was named after Peter Jackson by Soo-Jen, who was the guy who gave us the samples in the first place. In addition to that, there was a proposal not so long ago by Carlos Driscoll and Soo-Jen Liu to basically consider the restoration of tigers in a range of the Caspian tiger. The Caspian tiger lives in areas where the former Soviet republics are. It's as big as continental United States. It used to have a big range of tigers. There are none there now. Let's introduce some in there. The reason that they were able to say that is because it turns out that the genetic studies showed that the Caspian tiger and the Amur tiger were virtually indistinguishable, and we have hundreds of Amur tigers in captivity waiting to be introduced. There are more stories to tell you. I won't go into any more. Simply say that whole genomes of 10,000 species is being led by the Genome 10K project, which is an ongoing consortium of people across the world trying to sequence every species or vertebrate we can get our hands on. The Vertebrate Genome Project wants to do all 62,000, whether we can get our hands on them or not, to give to the next generation a whole genome sequence of every species that we can catch. These are some of the species that have been sequenced already. There are now 500 species that have whole genome sequences of vertebrates. In addition to that, there are derivative projects such as Insect 5K, the Rabbit Project, the Bird Project, and right here we have the leader of the Global Invertebrate Genome Alliance. This is a project to sequence tens of thousands of marine invertebrates, the sponges, the jellyfish. It's led by Joe Lopez. Raise your hand, Joe. He's right there in the audience. He's basically coordinated the collection and the coordination of the workers across the world so that the genomic data, like the ones I've talked about, can be applied to these species. Finally, the Earth Biogenome Project takes all of these, adds to it the invertebrates, the beasts, everything is over. And now I'd just like to say that there's a lot going on in the oceans that needs to be mentioned. This is my last slide, Bernard, so relax. The ocean covers 71% of the Earth's surface, 50% of the species live there. The phytoplankton supply twice as much oxygen as the Amazon, so every second breath you take comes from the ocean. Only 5% of the seas are covered with marine protected areas, and we can use science to indicate real action. Life can return with the proper protection and investments. So the whole thing is that we really can move ahead Genetics is not the answer to everything, but it is the answer to some things, and I think that I'm excited to be here at a Coral Reef Symposium talking to you about oceans because we're just starting to get into that. Now I'd like to say to the world conservation community, and thank you for listening, particularly Bernard for your patience.